opportunities, but she's successful at obtaining them, including the UREX Award, which is Oregon State's highest award for graduate level research that honors intellectual vitality along with academic excellence. I have to quote that. She also received an NSF GRIP fellowship. So that's Graduate Research Internship Program fellowship to work with our colleagues at the US Geological Survey down in Flagstaff for her Grand Canyon based research that she'll talk about. And most recently, she completed a US Fish and Wildlife internship with a group based up in Portland. So all of these are competitive fellowships and grants that she's been successful at obtaining. And that's just the beginning of the list. Now, beyond that kind of success, Erin has these amazing leadership skills. And in fact, I've taken note of some of her techniques and tried to incorporate that into how I run the research lab here. As you know, she was president of the Graduate Student Union for a year and has been highly involved in contract negotiations and bargaining at the highest levels of the university. In fact, at one point, she was having monthly head-to-head sit-down meetings with the president of the university, Ed Ray, and was just very casual about telling me about some of these meetings. It's just sort of an impressive position to be in for a graduate student. Erin is extremely organized. Whenever we have a meeting, she keeps me on track with what the agenda is and is very sort of organized and friendly about what she wants to accomplish, and she sticks to it. And, you know, just speaking to that, she was offered a postdoc with Elizabeth Anderson at Florida International University, which she's going to start in January, which essentially lit a fire under finishing the PhD. And I was skeptical. You could pull this off, Erin. But man, she just organized everything, pulled all the final data analysis and write up together, and I think has put together a really beautiful package that she's going to present to you today. And lest I make it sound like Erin is this like super effective grant writer, leader, organizer, which she is, she also has this amazingly interesting side where she's an excellent hunter, and man, she could tell you some stories about hunting that are just so crazy and exciting and fun. She's also an accomplished whitewater kayaker and swiftwater rescue expert, and she's really good at baking delicious treats. So unfortunately, there isn't probably going to be a face-to-face party where we can all share some delicious stuff that Erin cooks and enjoy a beverage together. There'll probably be a virtual version of this, but she's capable of that. And so hopefully someday we'll all get together and do some more of that. So with that, I want to turn this over to Erin. I could keep going on, but you know, I want her work and her to speak for herself here. And I'll turn it over to you, Erin.
Sweet. Thanks for that introduction, Dave. It's been a pleasure working with you over the last 5 years. I've really enjoyed being a part of the lab, and I'm forever grateful for having this experience with you in the lab. So before I start, I want to let folks know that I do have a cough, so I might be taking some water breaks, but don't fear, I tested negative for COVID after this cough started, so I'm attributing it to allergies. So to orient you to my talk, I will be providing this talk map with icons that will appear at the bottom of each of the slides. The first of my PhD projects that I want to talk about today examines who is doing the science. So I have it in scare quotes because it's only looking at scientific societies, whereas many, many different organizations and individual humans are doing science out in the world. Secondly, I'll be talking about the science that I'm doing, which examines how human alterations, specifically dams, impact aquatic organisms on the community and molecular levels. And to end the presentation, I want to talk about what I perceive as the biggest challenges facing humanity and how we should address them. I'd like to start with a land acknowledgement statement. If you've never heard of this before, land acknowledgement is a small step towards honoring the truth, because unless we are Native Americans on our homelands, we are settlers on stolen land. So for Oregon State University in Corvallis, Oregon, it is located within the traditional homelands of the Marys River or Ampine Fu Band of the Kalapuya. Following the Willamette Valley Treaty of 1855, Kalapuya people were forcibly removed to reservations in Western Oregon. Today, living descendants of these folks are part of the Confederated Tribes of Grand Ronde Community of Oregon and the Confederated Tribes of the Siletz Indians. I included this resource here, which shows Native American territories in North America, and they've actually expanded this resource to parts of South America, Australia, and I know they're working on expanding it globally. It's a searchable map, and as you go into the territories, it has links to websites for many of the tribes. So I encourage you to go here and learn about the original stewards of the land on which you live and work. And that website's native-land.ca. So one of the most rewarding projects that I've had the opportunity to work on is getting at this question of who is doing the science. And I, along with 10 co-authors, focused this project on a professional scientific society. So you might ask yourself, why investigating the demographics of a scientific society are important. Well, for centuries, science has been the near exclusive domain of upper-class white men, and there have been limited opportunities for marginalized folks. And while participation has broadened over the last several decades, most areas of science still lack inclusive and equitable participation by marginalized groups. So we have this opportunity to try and broaden participation, which I really consider to be a moral obligation to our fellow humans. But there is science to back up the need to do this diversity work as well. Diversity of membership promotes good science by broadening viewpoints, expanding the questions that get asked, and bringing different problem-solving skills to the table. It increases team functioning, innovation, and results in higher impact science. And then we focus on inclusivity in addition to diversity, because inclusion allows the benefits of diversity to happen. So inclusion welcomes and values folks with marginalized identities. So there's a need and there is good reason to do this work. We focused our project on the Society for Freshwater Science, which is an international scientific organization that seeks to promote the understanding of freshwater ecosystems. So that's rivers, ponds, streams. Its mission is to foster the understanding of information among its members and other scientific societies, resource managers, policymakers, educators, and the public. So basically everyone. And to the point of diversity and inclusion work, many SFS, Society for Freshwater Science members have worked throughout the years to increase participation of marginalized groups. And SFS leadership is generally very supportive of those actions. So this is a great society for our case study, and it's also my home scientific society. In undergrad, Dr. Mike Gengloff took me to my first scientific conference, which was SFS, and it was such a great experience that I couldn't wait to go back when I joined Dave's lab. I also met Dr. Elizabeth Anderson there, who, like Dave said, will be my postdoc advisor. And so this organization and its people are very close to me as my home scientific society. So for this project, which is now published in Freshwater Science, we completed a survey in 2017, and we had almost 300 responses, which was about 20% of the current membership of SFS. 66% of survey participants were under the age of 50, half had been members of SFS for more than 10 years, and most were US residents and worked in academia, so a university setting. It wasn't really a surprise to me, as I'd been going to these conferences for years, but SFS is also very white. It's 87% of folks identified as white. But the fact that 10% identified as LGBTQ+ was surprising, mostly because that's relatively high to the US population. And LGBTQ+ means lesbian, gay, bisexual, transgender, and queer. So to dive into this comparison of SFS demographics to the US population, to give you some idea, I invite you to look at this graph. So we have the percent of the population on the y-axis and the demographic groups on the x-axis. And I'm showing two data sets here. You have SFS membership in the white bars, and then the US population in the black bars. So SFS had a lower percentage of folks identifying as having a disability, lower percentage as those identifying as these racial minority groups and as women. But then for the folks identifying as transgender or otherwise LGBTQ+, we see a higher percentage in the survey respondents, higher percentage in the in the SFS population than the US population. So this result has two possible explanations, we think. First, LGBTQ members may have been more likely to respond to the survey, but it is also possible that SFS has a relatively large LGBTQ+ minority population. So for our project, we really wanted to highlight some of the actions that SFS is already doing to increase diversity and inclusion. And we're going to highlight the InSTARS program. So this program was initiated by members, and over the last 9 years, it has sponsored undergraduate students with marginalized identities to attend the SFS annual meeting. So we have an annual meeting. And here the leadership of SFS, the organization, allocates $10,000 a year to send 14 students to the meeting. And in addition to that financial support, they provide a pre-conference orientation and a graduate student mentor to really help these students have the best possible experience they can at this conference. So InSTARS has had 121 fellows, of which 90% continue to conduct scientific research after attending SFS, the annual meeting. So in our paper, we also talk about the opportunities for improvement. And one of our suggestions is to increase funding for and expand this program. And when I did my practice presentation, Dave cued me into this email I'd missed. And I want to tell you that InSTARS is getting additional funding from the National Science Foundation to grow over the next 5 years. And that's huge. That's just huge. And the new program is called Emerge. So if you think about instars are like baby bugs, and then they morph into adults that emerge out of the water. But we'll talk about insect life cycles later in the talk, and we're going to stick with humans for now. But I just want to emphasize how awesome it is that members of SFS were able to write grants and get that funding for the InSTARS program to expand. So SFS is doing some amazing things, but there have been some mistakes. So my co-authors and I want to acknowledge those and give context for why we started this project. So back in 2017, North Carolina passed House Bill 2, which eliminated anti-discrimination protection for LGBTQ+ folks and made it illegal for people to use a bathroom that didn't correspond to the sex on their birth certificate. So since SFS already had a deposit on the venue in North Carolina, the leadership decided to keep the location. They did craft a meeting inclusivity plan, but many members considered the location a barrier to attending the meeting. Some federal agencies wouldn't even allow their employees to travel to North Carolina. Our survey showed that LGBTQ+ folks that were members especially considered this law to be a barrier to attending the annual meeting. So we believe that the decision-making process here could have better incorporated the viewpoints of marginalized folks. But our survey and an ad hoc committee on inclusivity were grassroots efforts that resulted from the decision to keep the meeting in North Carolina. And through this ad hoc committee work, there were opening plenary talks on the issue of House Bill 2 and generally talking about diversity and inclusion in science. There were also 2 workshops on LGBTQ issues. Badges featured a place for you to write your pronouns, and gender neutral bathroom maps were provided. So all of this helped to make the meeting more inclusive for transgender and gender non-conforming members. And since this meeting, both the leadership of SFS and the membership have been working towards improvements. Our publication ends with recommendations for SFS to continue to improve, in addition to actions that individual members can take in their own lives. So I'll go into those now. Specifically, we suggest that SFS increase representation of folks with a range of marginalized identities. So identities, think race, ability, sexuality, parental status. And we also suggested they increase that representation within the society and at the meetings through society awards and the media that it produces. We'd like to see a better dialogue between leadership and members, specifically through creative formats. You know, it's really hard to just show up at a board meeting and express concerns. So thinking of creative ways to facilitate that communication is important. We also recommend ways to expand diversity efforts at meetings themselves. One example being affinity breakfasts. So this affinity breakfast could be SFS providing a space and some food or money for food for folks with specific identities to gather and connect during the meeting. Lastly, we recommend promoting the NSTARS program, which I'm happy to see is happening. We also wanted to speak directly to SFS members with our paper and remind readers that diversity and inclusion work really starts with you. You've got to educate yourself about the lived experiences of people with different identities who experience discrimination and exclusion. Importantly, you've got to try and avoid asking others to educate you. Instead, you can seek out available resources to educate yourself, such as following people on social media, like Black Birders Week is an example of one of the social media campaigns to raise awareness of people of color in science. And a super important aspect of this education is to take time to reflect upon your own social identity as well, and consider how various aspects of it may affect your privilege and benefits that you experience in scientific and public spaces. Many people feel that their identity is normal. However, this usually comes from being in a demographically dominant position in society. So if you have privilege, it's really important to consider how you can use that privilege to uplift and support others. And I found this book, What Does It Mean to Be White by Robin DiAngelo, to be very helpful as a white person doing this self-exploration myself. So to grow your efforts from that personal level, you can encourage open conversations about inclusivity and diversity in the groups you work with. It's really important to be aware of and actively work against power dynamics and internal biases that may limit these conversations you're trying to facilitate. And remember, I can't emphasize enough, to always promote and trust the voices of marginalized people. But before you actively recruit and mentor folks with marginalized identities, I think it's really important to seek out and training for yourself and your colleagues on how to be an effective mentor and advocate for people with marginalized identities. Along with this broad training, I really think you've got to understand the environments that you're bringing someone into. So academia can be a very unwelcoming place for people holding marginalized identities. At OSU, 20% of grads say that they're impacted by harassment at work. 20%, that's a lot of people. Most people don't work in a building that has gender-neutral bathrooms. And nearly a fourth of folks are spending the majority of their income on rent. If you have a better understanding of the working and living conditions of the students that you're taking on, you'll be able to speak honestly and openly with prospective students, and you'll be a more effective mentor when you're helping them get through those hardships during grad school. That will help with retention, and it will potentially motivate you to help fix some of those systemic issues that are disproportionately impacting students with marginalized identities. So I really like the analogy that if diversity is being invited to the party, inclusivity is being asked to dance. But we're in a lab, we're not at a dance party. So how do you know if folks are really feeling comfortable and included? Well, I think a really good metric is laughter. And I'm not alone. The ecologist Robert Payne is said to have claimed that he could tell which grad students would be the most successful because they were the ones having the most fun. If you and your lab and your department are laughing so hard that you can't hold it together, you know you're on the right path. In summary, diversity and inclusion work is valuable for society, for science, and it's a moral obligation to our fellow humans. There's really a need within our majority white, cisgender, heterosexual societies for this work, especially for this work to be done at multiple organizational levels and not just by the marginalized folks most affected. We provide resources in our paper, but these are things institutions need to incorporate into their core values and pay folks to work on. It was so rewarding to work on this project with these amazing humans from all over. I also want to thank our survey participants for speaking and SFS leadership for listening. I think this project is already helping push SFS in a great direction. And I have the publication there, information there if you want to check it out. Freshwater science. Okay, so that's kind of who is doing the science, but what science am I doing? Well, the work that I've done at OSU has revolved around rivers, specifically how dams impact aquatic organisms. People love rivers. As Dave mentioned, I'm a whitewater kayaker, but we also use our rivers. We use them for things like drinking water, irrigation, wastewater removal, and power. So I'd like to zero in on hydropower and the fact that humans dam rivers for this purpose. So we've got a kayaker here and then a hydropower plant in the background. Okay, so it's not like we just dam a few rivers here and there. Humans are very industrious. This video shows the location of about 7,000 dams, but there are closer to 57,000 dams, large dams worldwide. And that's just the dams that are over 15 meters. So the point here is dams are everywhere and they are impacting the majority of rivers on Earth. So what are those impacts on the river? Besides turning a large portion of the river into a lake, dams alter the flow of water, nutrients, sediment, and organisms. And they alter that in the tailwater portion of the river. So that's the river directly downstream of the dam structure. Specifically, you can see here on this graph how dams influence the flow of water. So we have gauge height on the y-axis, and then the days of the week on the x-axis. And you can see that in order to generate power at Hoover Dam, there is a change in the water height of 8 feet. That's like tidal changes at the beach. We call this hydropeaking. Hydropeaking is the variation in daily discharge. So you have a certain level in the morning and you have another level in the evening. The bigger the distance between those two, the bigger the hydropeaking index. So this type of flow alteration affects the structure of the biological communities. This graph is from a paper by some of my co-authors published in 2016. The tailwaters experiencing the high hydropeaking, so hydropeaking is on the x-axis, they have the very few insect taxa. So insect taxa, percent insect taxa is shown here on the y-axis. And these are specific insects. So they're EPT. So that's Ephemeroptera, Plecoptera, and Trichoptera insects. And the important element to remember here with those three is that scientists understand these insects to be sensitive. So whether that's sensitive to pollution or in this case, flow. So what do we care if there are fewer of these EPT insects? Well, these are generally the species in the invertebrate communities that emerge from the water as adults. So you can see the juveniles here in the water emerging as adults. And many of these baby bugs are providing food for aquatic organisms like fish, and then they emerge as adults and provide food for birds and bats and spiders. But the hydropeaking, as you can see here, we have dammed and then undammed plus hydropeaking. Hydropeaking impacts the types and number of aquatic invertebrates in the water that are available to consumers here in the water or on land. So break slide. So scientists have some theories, but many case studies on individual dams to help us understand how dams impact rivers. But there doesn't exist this unifying theory of tailwaters that could help us understand how these tens of thousands of dams are affecting our rivers in these tailwater areas, or how they're shaping biological communities. So that's what I hope to help kind of flesh out with this project. Our first research question is, how do hydropower dams influence invertebrate dominance and community structure? So that's essentially asking who is in the invertebrate community and are there a few major players? Secondly, does hydropeaking intensity affect the proportion of insect and EPT taxa? And third, how does dominance, richness, abundance, and biomass change with increasing distance from the dam? So increasing distance away from the dam downstream. To answer these questions, we sampled the invertebrate communities in the tailwaters downstream of 7 large dams in the Colorado River Basin, collecting samples at increasing distances downstream of the dam out to 25 river kilometers. Those community samples were taken back to the lab and identified. By identifying the invertebrates, we got richness, so how many species in each sample. And then we counted and we measured them. And that data allowed us to calculate the abundance and the biomass of each species. And from there, I calculated the richness and abundance and biomass for the whole community, so all the insects and then the sensitive insects, the EPT taxa, as well as dominance statistics and the percent EPT and percent insect. So results. We found that each tailwater was dominated by 3 to 7 invertebrate taxa that comprised 95% of all the individuals we collected. This top row is all invertebrates that are going to stay in the water their whole life. So a mussel, a snail, aquatic worms, and mites. Whereas the bottom row, they'll be in the river as young invertebrates, but then they'll morph into a flying adult later in life. So this has implications for where the resources are contained. The resources that are contained in their bodies will be utilized by other organisms. If you think about an aquatic mite, it's not very likely to feed a bird, but a flying insect like a mayfly might. So ultimately, many of these dominant taxa are non-insect and they're non-flying species and thus unavailable to terrestrial consumers, this whole top row. Hydropeaking did have the impact that we predicted. So when you have increasing hydropeaking, which is on the x-axis, we observed a decrease in EPT biomass. So that's the mass of those sensitive insects. The colored points here represent different dams that we sampled downstream of. And like we found with the dominant community members, a decrease in EPT puts limits on the type and quality of the food available to other animals in the ecosystem. So when you have less EPT, it means there's less food for organisms in both the water and in the surrounding areas. We also observed the striking positive relationship. So richness, which is the number of species on the y-axis, is actually genus, number of different genus we have, which is the next highest up taxonomic classification. So richness generally increases with increasing distance from the dam, which is on the x-axis. So this is showing us that even within 25 river kilometers downstream of the dam, insects are coming back into the system. So notable exceptions are Glen Canyon here on the bottom, which is a flat line, so there's no recovery, and Hoover Dam, which is negative. And that's likely because the part of the tailwater reach sampled down here is starting to pool up from the next reservoir downstream. So this recovery couldn't happen because we ran into the effects of the next dam. So the take-home messages with this chapter, while there are a lot of invertebrates downstream of these dams, it's only a few species. We found that hydropeaking did impact EPT, abundance, and biomass, negatively impact it. And then lastly, distance from dam also impacted EPT richness, abundance, and biomass, with all metrics increasing with distance from dam. So they got more so as you got farther away from the dam. So these findings suggest the impacts of dammings are most noticeable immediately downstream of dams, and then they decrease. But the big picture, our results demonstrate that Western US tailwaters, so those sections downstream of dams, can support aquatic invertebrate communities with high abundances, but low diversity. So many people helped with this project. I couldn't even fit all the pictures here, so I put names. I've included the co-authors on this paper, the USGS employees who collected the invertebrates, OSU students who picked the invertebrates in the lab, and folks who helped me write this publication through our journal club here at Integrated Biology. And this paper is actually in review at a scientific journal right now. It was also a super fun project to be involved with. So we know that dams impact flows of a river downstream, but there's a lot of other impacts as well. So for my second dam project, I focused on how dams impact population structure, which we can think about as organisms moving genes around on the landscape. Here we have a cartoon of a straightforward example of a dam blocking the upstream movement of fish. Those fish aren't going to be able to exchange genes with fish upstream, and that species will likely show some population structure between those two groups. One group's upstream and one group's downstream of the dam. Now, the up and downstream of a dam is a straightforward example. But if we think about my previous project and we start to ask the question of how dams create barriers completely downstream of a section of river that is dammed, you have certain insects that can't live in it. So if you imagine that there's a dam upstream of this circle that has made the main section of this river uninhabitable. So maybe the insect can live off in this side piece here, but the main section is made uninhabitable by the dam. How is that insect going to then move to this circle? Are they going to be able to? Or are these now two fragmented populations? So we're going to zero in on aquatic insects here, many of which have an aquatic phase as a juvenile called a larva that then morph into a winged adult. So that larva can get around in a few ways. They can crawl or swim up or downstream. They can also get swept downstream by the flow of the water. Adults can put those wings on and go to work flying to a new location, or they can float passively on the wind. The diagram here on the right is showing some of the routes that these insects can take. You know, some need to stick physically to the water, either in it itself or flying above it, using it as a cue. Others can just head off across land and hope they find water again. Then there's this idea of distance. Some insects can fly more than 1 kilometer, which is quite far for a tiny bug. And then ultimately, dispersal strength, which you can think about how far they go, depends on the body of the insect and the environment in which they live. So we're going to have a short genetics lesson here to help orient you to my hypotheses for this project. So the relationship between genetic distance, genetic distance here on the y-axis, or how similar organisms are genetically, and geographic distance, here we go, geographic distance, can provide insight into dispersal strength and possible evolutionary mechanisms. When a species exhibits high genetic distance, so very different, like in the first box, and high variance, lots of variation, regardless of geographic distance between sampling points, this indicates low dispersal ability. So we've got this first box with low dispersibility, and that evolutionary mechanism at work there is genetic drift. And then when species have low genetic distance, so the middle panel, and low variance, regardless of the geographic distance, this indicates high dispersal ability and gene flow. And gene flow is the evolutionary mechanism at work there. And gene flow is just the transfer of genetic material from one population to another. Oh, and I didn't tell you what genetic drift is. This lesson is lacking. So genetic drift is the random chance of that genetic change happening. So genetic drift is that random chance happening because it's low dispersal. Maybe the populations aren't able to connect. So this population might change by random chance or genetic drift. But then when you have that high gene flow, you've got the transfer of genes between populations, and you have low difference between the genes there. It'd be more of a mouse and a mouse situation than a mouse and a human. Okay. So then the last example, we actually have a difference. So you have a positive relationship between the genetic and geographic distance that indicates moderate dispersal ability. And we call that, or the mechanism here is isolation by distance. So that means that pairs of populations close to each other physically will be more genetically similar. And then as you get farther away in space, you're also going to get farther away in genetic similarity. Okay, so to tie it all together, you've got organisms with varying dispersal abilities using different tactics to try to move around across the landscape and find mates. Some of those landscapes present barriers, like dams that may render the river uninhabitable. But there are also natural barriers, like a desert or a mountain, that can be difficult to get around. So in the Grand Canyon, we've got all types of potential barriers. We've got the Glen Canyon Dam that makes the main stem practically uninhabitable for EPT insects, although many more insects are able to live in the tributaries. So tributaries are small streams coming into a main river. But those insects in the tributaries, how are they able to get, between these tributaries, or are they isolated populations? Because if you think about it, you've got that main stem that's highly altered, and they're in these canyons that are super deep. The Grand Canyon's 1.5 kilometers deep on average. So these high canyon walls could be a barrier to, tiny flying insects, which brings me to my hypotheses. So number one, we predict that the relationships between a species' genetic distance and geographic distance will be mediated by adult dispersal ability. So basically, we expect to see the patterns that I described before. And then number two, we predict that for species most heavily influenced by isolation by distance, that Euclidean or straight line distance would be the best predictor for would be the best predictor, or be a better predictor than river network distance. So if we can get at this, it would help us understand the question of whether human or natural barriers are at play. So in essence, if river network distance, so this distance between the 2 points as traced by the river network, is more informative, it's likely that the main stem isn't a barrier, and that the deep canyon walls are acting more as a natural dispersal barrier. But if the straight line distance there is more informative, that would suggest that insects are dispersing up and over these steep canyon walls, and that maybe the main stem of the Colorado is the potential barrier. Okay, so who did we choose to test these hypotheses? We chose three species, a water strider called Ragovelia distincta here, and a caddisfly, Hydrocycid osleri, and a mayfly, Falcion quelleri. I've shown photos here of a larvae and an adult stage. The water strider kind of looks the same in both, so that's why there's not two pictures. These represent low, high, and medium dispersal abilities based on research into these species' physical traits and abilities. Luckily, this research was in the scientific literature, so I didn't have to do it myself. I'm using genetics to test their predictions. So the literature I used it's called trait database literature, and it's a really deep area of scientific research. Folks use these databases to collate information about species from many, many different resources. And we can then use these traits to understand, individual ecosystems out on the landscape. So say you're in an ecosystem and you find all these insects that love rivers that just dry up. But whenever you go to this creek, it always has water in it. So knowing something about the traits of the organisms that you find there can help you make predictions about that ecosystem. So maybe you visit that stream a little more frequently to see if it does indeed dry up. So to collect these three insects in the Grand Canyon, I rafted down the Colorado River with the U.S. Geological Survey and Grand Canyon Youth, which is the nonprofit, or a nonprofit, the nonprofit that takes high school students down the Grand Canyon. So we stopped at tributaries, which again are those small streams that are flowing into the Colorado River, and we collected insects. We only collected at the tributaries because previous research had already shown us that there's very few insects in the main river. And it was our goal to get more than 12 of each of these insects. I think we usually got about 30 whenever we could find them. And then we euthanized them by storing them in ethanol. And then we stored that ethanol in coolers to help preserve them before we could get back to freezers, because we were in the bottom of the Grand Canyon. So back in the lab, we identified the insects down to species and extracted the DNA. So I used a genetics protocol that identifies small portions of their DNA. So you already had one genetics lesson today, so I'm going to spare you the coding details for how I filtered the genetic sequences. But I do want to give you some theory on how this works. So if you remember, DNA is made up of A's, C's, T's, and G's. And we got thousands of the small portions of the DNA that we identified. So when we sequenced it, we've got a lot of those small portions. So within those small sections, we were the things we were ultimately looking for. So what we were looking for in those small sections of DNA were called SNPs, single nucleotide polymorphisms. So if you remember, nucleotides are those A's, T's, C's, and G's. And so we were looking for, let's see, a SNP, a single nucleotide polymorphism, is we're looking for where those A's, T's, and C's, and G's differ across multiple individuals. So in this case, one insect may have an A and another may have a G, and we use this information to tell us how closely related the individual insects are. So if you glossed over all that A's, T's, C's, and G's, don't worry, remember this. The more similar the SNPs are, so the single nucleotide polymorphisms, the more closely related the individual insects are. So to get into that SNP data, which is what we ultimately wanted from all this genetics work, that's where we calculated our genetic distance. So that's here on the y-axis again, and the geographic distance is on the x-axis. So remember that we predicted that the water strider would be low and the caddisfly would be a high disperser? Well, it turned out that they're both high dispersers based on the genetics data. So we have low genetic difference and low variance based on these trend lines. So low genetic difference here, low, and low variance. So they're both high dispersers with the evolutionary mechanism of gene flow at work here in the Grand Canyon. So that mayfly, Falcion quelleri, that we extracted DNA from, we predicted that would be a moderate disperser showing isolation by distance. And lo and behold, we saw it with the genetics data, which was super interesting. So with increasing genetic distance, or with increasing geographic distance, you also have increasing genetic distance. So to get at that second hypothesis, you know, whether Euclidean or straight line distance is more informative or river network distance, I ran models to see which was more informative, and it turns out there was no difference. So unfortunately, that method didn't give us much insight on the difference between human versus natural barriers. But something interesting that we did notice was that with caddisflies, there was some separation between populations located on the north rim versus those on the south rim of the Grand Canyon. So what we're looking at here is a principal components analysis. Basically, it's taking the genetic information and arranging the points, which are individuals in 2D space, where those that are genetically similar are closer in space on this graph. So when we sort the individuals by those that are on the north versus the south rim, we see this separation between the two groups. And I think this has something to do with the natural geography of the Grand Canyon, because this is a high dispersing species. Remember, I'm working with this idea with my USGS colleagues who published this paper, and they've got this unique perspective because they published this paper this year in which they saw separation with the same species between the east and the west parts of the Grand Canyon. But they attributed it to the Toroweap Fault. So if you remember from geography class, a fault is a fracture between two blocks of rock. So we're still brainstorming about this, but to me it looks like natural geography may have created this division between populations. So in conclusion, water striders and caddisflies are high dispersers with high gene flow, and mayflies are moderate dispersers showing isolation by distance. So the fact that water striders didn't match our hypotheses is a good reminder that sometimes realized dispersal ability may not be accurately predicted by trait databases. And for our second hypothesis about whether human or natural geographic barriers are more influential, we didn't get good resolution on that with our methods, but there is without a doubt this system is highly altered. So if you raft down the Grand Canyon, you'll constantly be aware of these artificial tides that sweep down the Colorado River from Glen Canyon Dam. The fact that it's uninhabited by most insects is a signal that something is going on here. But how that impacts dispersal, we're still trying to figure out. So what we did see clearly was that division between the North and the South Rim caddisfly populations, and likely that's some combination of the geologic barriers like the Torweep Fault acting in tandem with the main stem of the Colorado River being altered that has created this population division. The big picture here, this type of genetics data can be used to help inform effective conservation management plans for understudied organisms that are also organisms of conservation interest, like aquatic insects. So I can't thank enough my co-authors, the USGS employees, and the high school students and RAF guides and Grand Canyon youth coordinators who made this work possible. It was a joy, just an absolute joy. If you know any high school students, sign them up for these trips when they start running again. They are an incredible experience. The kids don't have to want to go into science. They're not trying to make scientists. They're just trying to get them out in nature and experience doing science, interacting with graduate students. It's just an absolute joy to be a part of this program. And I recommend everybody to look it up and send high school students that way. So as I finish this presentation, I wanted to take a step back and provide some context for the motivation behind my PhD and future postdoc work. I joined Dave's lab very interested in human impacts on freshwater ecosystem. And as I started the research, I became more and more interested in the human aspect than I could have ever expected. I was becoming more and more aware of the whiteness of the professional societies that I was a part of, and how the experiences of my fellow graduate students of color differed so greatly from my own at Oregon State University. And as I worked on the SFS project and got really involved in my graduate student union, I started looking at more closely at the living and working conditions of grad students. So when I think about the three biggest challenges to humans right now, I think COVID-19, climate change, and just systemic racism. And these might seem like overwhelming challenges individually, and even more so when you think about them together. But within this PhD program, I think I've really discovered how humans must operate in order to overcome these challenges. And the answer, my friends, is collective action and collective power. I have never seen the will of the people enacted so quickly as I saw during collective bargaining with the Graduate Student Labor Union. And you might say, Erin, was it quick? And it's okay. It took us time and it took us a lot of people energy. Over 9 months with a core team of 20 and the support of hundreds of graduate students, but we won tangible, substantial change and benefits for grad employees. We won raises and paid parental leave, even as the pandemic was sending us home to work and to bargain. To address the concerns of being forced to return to work during the pandemic, grads returned to the bargaining table and got protections on how to return to work and got OSU to agree to cover home internet costs for grad workers. But the living and working conditions of grad employees aren't confined to the workplace or how much we make in a paycheck. Systemic racism is a pervasive problem in our society that people are using collective power to overcome. In our department this year, grads were able to motivate the integrative biology leadership to remove the GRE requirement. So the GRE is this entrance exam that is still required by many graduate programs. It's expensive, and students of color consistently score lower on it. It's just this terrible metric for determining who you should allow into graduate school. But collective power removed it within this department, and collective power is getting it removed elsewhere. Additionally, at OSU, the Disarm movement is bringing together undergrads, grads, community members, and politicians to pressure OSU to not create its own campus police force. We simply don't need a police presence in our learning environment. And this group's also providing information on who they're hiring for the police force, who they're hiring for the leadership of it, and giving us information on those folks' backgrounds. Collective power is incredible. So big picture, to fight these three big challenges, it's going to take hundreds and thousands of these grassroots efforts. I'm not as involved in fighting climate change as I soon will be, but when I read about the collective actions of the Sunrise Movement, I have hope. So if you are concerned about any of these issues, I encourage you to figure out how to increase collective power to address them. You will be more powerful, you will feel more supported, and you will help humanity. So what's next for Erin Abernethy? Well, I'm leaving my department, Integrative Biology, with a Legacy Award. So it's an annual award of $500 distributed randomly to a graduate student in integrative biology. You can see my website for details, but I really want to help a fellow grad make rent 1 month so that they can focus on more important things like graduating and achieving their dreams, whatever they may be. This month, I'll be starting a postdoc with Elizabeth Anderson, based out of Florida International University. But given the pandemic situation, it's going to be a remote position. But the work I'll be doing will examine how race and gender influence water management in Florida, and how the incorporation of diversity work can be improved. And finally, I'd like to plug this program. Dave mentioned it in the beginning, but this program is just amazing. It's with the U.S. Fish and Wildlife Service, and I was an intern at the regional Portland office this past summer as a directorate fellow with Paul Heimwitz, and it was a simply amazing program and an incredible organization to work with. I highly recommend the program. So it's Directorate Fellows Program. And I recommend it to any graduate student who is considering a conservation career in federal service. Upon graduation and completion of this program, you get direct hiring authority, which is amazing and something that I very much look forward to utilizing with the U.S. Fish and Wildlife Service. So I am just beyond thankful to my committee, Dave, Molly, Alex, Desiree, and Jean, for your support over the years. My co-authors that I thank throughout the presentation have just been phenomenal. I don't understand how people publish alone. Co-authors are amazing. And I've thoroughly enjoyed working with all of these, so many wonderful people, all these wonderful people. So my friends are the best. They support me and tease me as needed. You reel me in and amp me up. And I have this wonderful, vast friend collection that I've been cultivating over the years, and they are just absolute stars. And I can't wait for the many years of adventures and collective actions to come. I'll just let you stare at those photos for a while because I know they're embarrassing, some of you. So speaking of collective action, here's that photo of the grad union bargaining team again. We spent 16 hours on our last bargaining call together, and it was epic and powerful. And I'm forever changed for the better for my interactions with my CGE community. And finally, family. I love my family so much. Yvonne, Robert, Garrett, and Carly are my favorite people in the whole wide world. You have given me all the support I have ever needed, and I know that you will always give me that support no matter what. Garrett and Carly, you are my wildest adventure buddies, and I can't wait to see what adventures we get up to next year. Mom and Dad, you prepared me for this life since the day I was born. So I'd actually like to read a prediction that my father wrote in 1989 when I was 4 months old. So here's the photo, and he wrote it on the back of the photo. So, a serious girl already concerned about global warming, but no matter, for in 2009, while just a junior at Cornell, she discovers a rare combination of salts and enzymes that together combine to produce electricity when exposed to sunlight. Instant energy and no pollution. For this remarkable discovery, Erin Foster Abernethy receives the Nobel Prize. Ta-da! So there are a few elements of this prediction we're off about, or perhaps are not realized yet. But ultimately, you predicted 31 years ago that I'd be here, and you've given me the constant support that I've needed and has helped me fly through the PhD program and life. Y'all end every phone call with "I love you" and "I'm proud of you." And I'm very, very lucky and thankful to have you as my parents. So thank you to everyone who attended the talk and has given me support over the years. I appreciate you all.
Thanks so much for that, Erin. I'm tearing up here a little bit. Oh, that was a really, really moving presentation. Wow. I'm seeing a lot of stuff showing up on the chat, giving you high fives and congratulations. I'd love for people to put in questions on the question and answers so that we can explore some of this research a little more deeply. I think there's one up there already. Erin, I'll let you, I'll sign out here and just let you choose which ones to take on.
Okay, yeah. I see Claire asked, how did you get such high participation? Okay. With the rate with the SFS survey. What did we do? We sent it out, so we have all the members' emails. And so I think we sent it out through the official, probably like listserv that the membership had. Because I don't think we had it set up at that time to be done when you registered for the conference. I think we sent it out through email beforehand. Oh, we did, because I remember I gave edits on it because it was an online survey. But yeah, I think when we set it up, I think it's a demographic survey that'll be continued to be done. And Anna's actually working on it this coming year. But I think it's going to be now tied into an option for putting in demographic information or answering questions when you register for the conference as well. Okay, so I'll try to start. Oh, maybe I should start at the top. Okay, I'll start at the top and go down. I saw one of the major species found in section 2 was the quagga mussel, and I know they're present in Lake Mead and other western reservoirs. Just wondering if Erin saw any correlation between invasive species presence and abundance, such as quagga mussels and EPT taxa and other native species like you saw that you'd like to see in the Colorado River. Just thinking about how invasive species and other anthropogenic disturbances in these systems. Yeah, so we saw quagga mussels and zebra mussels, and I didn't look at correlations. Like I didn't try to correlate the numbers of it, the richness, numbers of like abundance or richness of the invasive species we found with the aquatic insects. I think because we just found those like two. I think it was just those two that were the invasive species invertebrates that we saw. But yeah, there was definitely downstream of Davis Dam, I think was the most, and it was just there were a high number of the quagga mussels. Jamie, really enjoyed your talk, Erin. The picture on your take-home slide of the Colorado River makes me wonder about the baseline aquatic insect abundance can be expected in rivers that are undammed in similar habitat. That's a great question, Jamie. So upstream of Glen Canyon Dam, once you get past the reservoir, you can go up into, Cataract Canyon and the Green and Colorado come up there, and they're a bit farther from dams. So they're kind of a more of a, maybe they give a better of an undammed example. And they do have aquatic insects. So there is evidence from other large river systems like this that aquatic insects can live in these large dynamic river systems. What governing body manages dams in the US? Okay, so I learned about FERC relicensing at my Fish and Wildlife internship. So like you definitely have to be licensed on the federal level for the dam, but then that would provide some constraints on how you operate your dam. But I think it's a much more local, like I would attribute the local decisions like this being much more important. I don't even know if it would be state specific or just kind of like dam specific, and then that dam operator, whether it's the Army Corps of Engineers or Western Power Authority, and they have to coordinate with their stakeholders. So it's definitely how a dam gets operated is a complicated measure. But I believe, I mean, you could probably say it's more dam specific, although there are, regulations at the state, regional, and federal level. How do you coordinate across the country if they're not coordinated? Yeah. Collective action. I don't know. It's definitely a complicated question, and it's one of the reasons that these issues are, that there's not a lot of research that like looks at the national level on this. High participation. Yes. Yep. I think we got such high participation from the SFS survey. And also I think there was high participation because there was a lot of emotions tied to that meeting. So people knew about the issue of HB2, the House Bill 2 that said you couldn't use a bathroom that didn't correlate to your birth certificate. And people wanted to provide feedback on that. Great presentation, Erin. Very inspiring to a first-year grad. I was wondering what it was like making those suggestions to SFS and if they were receptive to the recommendations. Yeah, it was really amazing. So a couple of the co-authors are very involved at SFS. One of the co-authors is in charge of the InStars program. We had a couple co-authors that are very involved with the board for SFS. And so these recommendations were taken and were brought to people and they weren't defensive, I guess is like the main thing. They were receptive to them. We also, when we were writing our paper, we talked to past presidents of SFS and we said, hey, we're working on this project. We'd like SFS to be aware of it and be receptive to, disseminating it to leadership as well as members. And they were very supportive of that. And then actually the most recent SFS president gave us funding for these affinity breakfasts. So if we'd had the meeting in person last year, which it was canceled because of COVID, me and Amy Roseman were setting up these affinity breakfasts. She was providing money for the space and the food for them to happen. And so that was super exciting. And then Anya, any evidence of north-south rim genetic differentiation for striders and mayflies? No. We saw it in the caddisflies. We need to talk. That's from Anya. She was the one that wrote the paper with the torweep fault. So I need to chat with Anya some more.
So Erin, in the interest of giving you a breather and also allowing people to leave that want to leave, I guess you can just click off anytime you want, but and keeping things on track, do you want to pick another question out of that list?
Oh my, there's so many people.
The list got to keep growing. You guys are awesome, but it's probably too much to tackle all at once here.
Okay. I guess I saw this one short one. How did you get involved with the U.S. Fish and Wildlife Service? So I saw an announcement on Twitter for the Directorate Fellowship Program, and then I applied and I got connected through that. So it's the deadline for that's coming up in January. So you still have time to apply if you are a graduate student. Yeah, I definitely recommend Googling it and finding the application information and then figuring out which they list all the different positions online. So you can kind of look through and find one that's of interest to you.
All right. Well, I'm going to applaud. At least you hear me. And now I suggest we take a short break and then we'll reconvene with your committee for the closed door part of the defense. And Erin, do we have a separate Zoom link for that, or how do we want to do that?
You should definitely have a separate Zoom link for that.
Okay, I can email out to the committee my Zoom room and we can go there.
Okay, well, why don't we do that? And do you want to reconvene at, say 11:15?
Sure, that sounds great.
Okay, great and awesome. Nice talk again, and we'll see you in just a minute.